Hey, welcome back to biology class. We are moving into chapter 24. In this chapter, we're going to talk about the cell cycle and mitosis. So even though we're not technically in the cancer chapter, it still is pretty relevant to how your cells control their growth and how problems here can lead to the development of tumors. To begin with, we're going to talk about what occurs in the cell cycle. Then we'll come back and talk about how it's controlled and some places where things can go wrong. So as a cell is growing, it's making proteins, it's getting bigger and bigger. Eventually, a cell can only get so big until it's not really going to be functional. So as the cell grows, it is also going to divide or split from one cell into two new cells, to two daughter cells. To do that, you have to be able to accurately copy the DNA or DNA replication, and you have to be able to accurately divide that DNA between the two daughter cells. And to do that, the cell is going to pass through the stages of the cell cycle, which is going to involve mitosis and cytokinesis. I'm hoping that most of this first part of this chapter is a review to you. You did take Biology 1 already, so you have talked about the stages of mitosis. You've talked about what cytokinesis is. We're going to go a little bit more in depth, maybe introduce some new concepts and ideas along with mitosis, which should be a good foundation that you have already. The cell cycle starts when two new cells are formed from that parent. So it goes from all these cells, how the cell goes through and copies into two. Then it starts all over again. One of these cells is going to go through the stages again. M phase is when the cells actually divide. So these are all the stages of mitosis. This is the M phase. This is going to happen nucleus first. So these are the stages of dividing the nucleus. Then cytokinesis is the stage of dividing the cytoplasm, or pinching that cell into two pieces. So at the start of mitosis, your chromatin is going to fold. It's going to condense, kind of nicely wrapping up your chromosomes so that they're not just floppy, hanging around, easily broken. They're condensed and folded and very neatly able to be divided. When your prophase has started, you actually have visible chromosomes. You can look in a light microscope and actually see those chromosomes. This is going to be after interphase. DNA is going to be replicated during the S phase of interphase. So each chromosome during mitosis is composed of two sister chromatids. Those sister chromatids are two chromosomes, identical, copied from each other, and still connected to each other. And then the microtubules, which are part of the mitotic spindle, are going to take those chromatids to opposite ends of the cell. So those two sister chromatids will separate, go to opposite ends, form into two new nuclei, and form into two new cells. During the whole cell cycle, very little of it is actually spent in the M phase. Most of the time that a cell is going through the cell cycle is going to be an interphase. Interphase is when the cell is able to function and do a lot of its jobs that it needs to be doing. In interphase, you have G1, when the cell is growing, just doing its cell jobs. S phase, when DNA is replicated. And G2 phase, when the cell is growing and preparing for mitosis. How long it takes a cell to get through a cell cycle is called the generation time. Different types of cells, different species will have different generation times. But in mammals, in culture, it takes about 18 to 24 hours. And even in your body, it probably takes about 24 hours to go through the cell cycle. Whether your cell is a healthy, normal cell growing correctly, or even a cancer cell, it's still going to be close to this 18 to 24 hour time span for a cell to divide, to go from one cell to two, and that is your generation time. G1 is going to be very variable in how long it lasts, depending on different cell types. G2 tends to be a lot shorter and less variable. Part of that is because during G1, a cell has to decide if it's going to divide again. So sometimes cells spend some time in G1 waiting for a signal, or just opting out of the cell cycle altogether. Cells that arrest in G1, cells that are waiting for a growth signal that aren't dividing actively, are in G0. A cell can go from G0 back into G1 once that growth signal comes and start dividing. Some cells will opt out of the cell cycle altogether, go into G0 permanently. And those cells that have opted out of the cell cycle altogether are terminally differentiated. So something like your nerve cell is not going to divide again. It has undergone terminal differentiation. So then following G1, the cell goes through S, copies its DNA, goes through G2, and then goes through mitosis. Those 2 copies of the chromosome that you made during the S phase are going to have to be distributed to those 2 daughter cells. Each daughter cell is going to need an accurate copy of the entire genome. So that is going to occur during M phase, which includes both nuclear division or mitosis and cytoplasmic division or cytokinesis. Mitosis is then going to be subdivided into 5 different stages. Prophase, here's prophase in an animal cell, prophase in a plant cell, prometaphase, plant and animal, metaphase in an animal and a plant, anaphase in an animal and a plant, and telophase in an animal and a plant. So in these lectures, I do have a few slides thrown in here of how we're doing mitosis in a plant cell. Although this class really focuses on animal cells, so while I do think it's interesting to see how the cells look different between a plant and an animal, what you really need to focus on is the animal cells. I won't actually ask you plant cell questions on your quizzes and exams. So the first stage is prophase. So as your DNA has been replicated in your S phase, it's gone into G2 phase. In G2, it's starting to get ready to go through mitosis. At the end of G2, chromosomes are starting to condense. And then prophase, chromosomes are condensing more. So there's not a sharp transition of here is G2, here is prophase, because your chromosomes are just condensing through that whole process, going from these diffuse chromatin of interphase to these very sharply defined chromosomes of prophase. But that technical definition of prophase is when individual chromosomes are visible in a light microscope. So if you are looking in a light microscope and those chromosomes are visible, it is definitely in prophase. And if they're not visible yet, it's not yet in prophase. In prophase, each chromosome is going to be made of 2 sister chromatids. So here is 1 chromosome, 2 sister chromatids. Here is 1 chromosome, 2 sister chromatids. Here's a chromosome, so 2 chromatids, 2 sister chromatids. And they're tightly attached to each other. You can see this. Each of these are tightly condensed, tightly attached. In your animal cells, your nucleoli are disappearing. So here's the nucleolus of an interphase cell. Here's the nucleolus disappearing in the prophase cell. It's still a little bit visible. It can be visible here. But your cell is not really doing a lot of protein creation at this stage. So it doesn't really have the need of a nucleolus to be around forming that ribosomal RNA. Your centrosomes are starting to be functioning. They're typically duplicated at the S phase, and during prophase is when they're starting to move to opposite ends. So here you can see, here are the centrosomes with the paracentrioles in them, and they're moving to opposite ends of the cell. Those 2 ends of the cell are going to be where your 2 cells are going to reform. Your centrosomes are forming a microtubule organizing center. So they're going to be what is going to organize all of these microtubules. These are the mitotic spindle that are made of microtubules, and even these little asters are made of microtubules. We're going to come back and talk about what each of these are doing. For now, they are being organized by the mitotic spindle. Within your within your centrosomes are these centriole pairs. Kind of look like a little orange Twizzler, like little snippets of a Twizzler here. And those little things are called centrioles. They're usually 2 working together at like a 90-degree angle from each other. Even as they're moving here, you have 2 of these little centrioles here. And this starburst shape of our microtubules around each around each centrosome is called an aster. They're forming an aster here. The next stage is prometaphase. During prometaphase, the nuclear membrane has degraded. So you no longer have a nucleus here. Your chromosomes are out, and that means that they are able to connect to the microtubules. So the centrosomes have moved to opposite sides of the nucleus, and they're connecting to different microtubules here. And the spindle microtubules are going to connect onto the chromosomes. The microtubules attach to the chromosomes at a region called the centromere, and they attach with something called a kinetochore. So these are your chromosomes. They have a centromere, that's part of the chromosomes. Chromosomes connect to the kinetochore. Kinetochore is attached to the kinetochore microtubules, which are going to help pull the chromosomes into their positions. So kinetochores start attaching to the centromeres shortly after S phase is completed. And during prometaphase, the spindle microtubules are going to attach to the kinetochores that are associated to each chromatid. So you have your chromosome with their chromatids connecting to the kinetochores, which are here in red, connecting to the microtubules, which are here in green. And those kinetochore microtubules are going to exert forces, pushing and pulling, to move the chromosomes into the center of the cell, right in between both of those, these spindle poles here. There's an inner kinetochore and an outer kinetochore. Here, this is the inner kinetochore. This is the part that attaches to the chromosome. And the outer kinetochore is the part that's going to attach to the microtubules. So inner kinetochore has proteins that bind to centromeric DNA. Outer kinetochore has proteins that attach to the ends of the microtubules. There's also something called a fibrous corona. So here's a chromosome, and you can see at this end it's attached to a kinetochore. Here it hasn't attached to anything yet. While that chromosome is not attached to a microtubule yet, or is not attached to a kinetochore yet, it forms what's called a fibrous corona. And that fibrous corona is going to be visible when the kinetochore is not attached. A mature kinetochore has two main parts, the constitutive centromere associated network, or CCAN, and the KMN network. The CCAN is going to be the centromere associated, that inner part of the kinetochore, and it has something called SNP and other proteins that attach it to the chromosome. The KMN network is the outer kinetochore. It stands for three different proteins that their initials are K, M, and N. And that is going to be what interacts with the microtubules. KMN is also able to interact with the CCAN, which is binding the centromeric chromatin. So that whole gap from chromosomes to microtubules are going to be connected to each other, from the inner kinetochore to the outer kinetochore to the microtubule. There are other kinds of microtubules besides the ones that are going to interact with the chromosomes. There are polar microtubules. Polar microtubules go from one spindle pole and then interact with microtubules that are coming from the opposite spindle pole. And then there are astral microtubules that are those short little starbursts that form those asters at each pole. So if I go backwards, here's your aster microtubules. And then, let's see. These would be a polar microtubule. It's not connected to a chromosome. This microtubule interacts with a microtubule from the other pole. Then our next phase is metaphase. In metaphase, you have fully condensed chromosomes, and they line up right in between your two poles. So you've got this nice line of chromosomes. That's called your metaphase plate. There's not an actual physical structure there. It's just kind of an imaginary line that creates your metaphase plate. And this is equidistant, or halfway equally between your two poles here. If you interfere with the spindle function, you can actually arrest the cells at this metaphase stage. So a drug like colchicine can make those cells get trapped here. During metaphase, it is these microtubules that are actively pulling the chromosomes toward opposite poles that is going to create that lining up at the metaphase plate. Anaphase is the shortest part of mitosis, and that is when chromosomes separate. So here you can see sister chromatids have let go of each other, and they're being pulled to opposite poles. There's two things that are going to happen here. Our kinetochore microtubules get shorter. That's anaphase A, when kinetochore microtubules get shorter. That pulls your chromosomes to opposite ends. In anaphase B, the polar microtubules get longer. That is going to pull these two poles away from each other. So anaphase A, chromosomes moving because the microtubules they're attached to are shorter. Anaphase B, poles separate from each other. The cell gets longer because the polar microtubules are getting longer. In some cells, anaphase A happens and then B. In some cells, they can happen at the same time. In telophase, you have daughter chromosomes at each spindle poles, and they're going to start to go back to being a normal functioning cell. So you're going to see chromosomes start to uncoil, go back to being that interphase chromatin. Mitotic spindles are disassociating. Nuclear envelope is reforming. You're starting to get a new nucleus around your chromosomes. And during this phase, sometimes even during the end of anaphase, during telophase, or sometimes following telophase, is when cytokinesis is going to start to take place. So you can see these cells, the membrane is starting to pinch in, forming that cleavage furrow that eventually gets tighter and tighter until those two cells pinch apart. Okay, so we're going to come back in our next video to talk a little bit more about how our mitotic spindle is moving, how it is controlling a lot of our chromosomes in our next video. So I'll see you in a little bit. Bye.